Hello, my name is Isabella Corsaro Alvarenga. I'm a PhD candidate in pet food science at Kansas State University, and today I'll be presenting a model that predicts corn-resistant starch type 2 during extrusion processing of a dog food kibble. Raw crystalline starches and less damaged starch granules are more resistant to enzymatic digestion in the animal. Previous research has shown that lowering mechanical energy of the extrusion process results in a greater retention of resistant starch. Resistant starches benefit animal health due to their prebiotic function, whereby the starch escaping digestion in the small intestine can be fermented with the production of butyrate, which is a short-chain fatty acid beneficial to the colonocyte. Based on previous work, we hypothesized that large starch particle size, low mechanical energy, and excess in-barrel moisture would lower starch gelatinization and yield more resistant starch. The study objective was to create a model that predicts resistant starch from corn in a dog food kibble by varying corn particle size, in-barrel moisture, and extruder chef speed. A single adult dog food recipe with 65% whole corn and no fiber additions were developed. Chicken meal served as the principal protein source, and no other fiber sources were included. Extrusion was performed in a small-scale twin-screw extruder using a central composite experimental design with 6 central points and 14 single replicates. Corn particle size, extruder chef speed, and in-barrel moisture were tested at 3 levels each, and all other variables were kept constant. Resistant starch from samples generated was measured with a kit. A surface response model to predict resistant starch in the resulting kibbles was developed using the REG and RS-REG procedure from CES. 3 plots were created for visualization. The first graph on the top left shows predicted resistant starch plotted as a function of chef speed and particle size. The plot on the right depicts resistant starch as a function of in-barrel moisture and particle size, and the plot on the bottom depicts the relationship of resistant starch as a function of in-barrel moisture and chef speed. The larger particle size, higher moisture, and slower chef speed resulted in the greatest resistant starch. This confirmed our hypothesis. A coarser grind appears to lower starch gelatinization due to a reduced surface area to mass. The lower mechanical energy may have allowed less energy transfer to cook the starch, and greater proportions of water likely acts as a plasticizer and which also decreased dough temperature. Rapid visco analyzer confirmed the starch transformations. The first plot, in this case an example for sample 15, was produced at low mechanical energy and high moisture. There was little to no starch damage due to mechanical shear, and the viscosity peak between 4 and 10 minutes indicates that there was raw starch still present in the sample. Conversely, sample 16 on the right had a larger initial increase in viscosity, which meant that there was extensive starch cooking by mechanical shear and little to no raw starch. Resistant starch content was 0.9% in sample 15, while sample 16 only had 0.35%. A model to predict resistant starch was created with the main effects of particle size, chef speed, and in-barrel moisture, and negative cross product between chef speed and particle size. Particle size could have greater effect if more separation had been achieved. We foresee challenges transferring this model to different extruders. We would like to acknowledge Hill's Pet Nutrition for sponsoring this research and the Grain Science Department at Kansas State University. Thank you.